Hi, and welcome to the Neurological Foundation's Discovery Sessions. The Neurological Foundation has been funding research into neurological conditions and disease for nearly 50 years. We have funded some amazing scientists and clinicians throughout their career who have gone on to conduct groundbreaking work and discover fascinating things about the human brain. We hope that these digital discovery sessions will help all our followers, supporters, and fellow science enthusiasts learn new things about one of the most complex organs humans have, their brain. I'd like to introduce today's presenter, Professor Maurice Curtis, the Deputy Director of the Neurological Foundation Human Brain Bank and Head of the Department of Anatomy and Medical Imaging at the University of Auckland. Professor Curtis has done groundbreaking work on the olfactory system, which was published in the world-leading journal Science. He has received a Miller Postgraduate Scholarship and Wrightson Postdoctoral Fellowship from the Neurological Foundation that has assisted him on his career path in neurological research. We're grateful to Professor Curtis for taking time today to give us a glimpse at a behind-the-scenes look at what the Neurological Foundation Human Brain Bank does and how it helps scientists in New Zealand and internationally conduct their research.
Good afternoon, everybody, and thank you for coming to listen a little bit more about the Human Brain Bank, the Neurological Foundation Human Brain Bank at the University of Auckland. Today, it's a pleasure to give you a little bit of a background to why we do what we do at the Human Brain Bank, how we go about processing a brain at the Human Brain Bank, and also how people come to be involved. How is it that they can sign up to be a brain donor for donating their brain to science? Then I'd like to tell you a little bit about a couple of interesting areas, I think, as to how the brain tissue can be used. Now, this is not meant to be exhaustive, it's just to give you an indication of how we need to preserve brain tissue differently, depending on what kind of a process we want to do scientifically with that tissue. So I'll start with the presentation now, and we're very grateful to the Neurological Foundation who have supported the work of the Brain Bank since 1994. And it was at that point it became the Neurological Foundation Human Brain Bank. For those of you that know anything around the Auckland Hospital region, we're located opposite Auckland Hospital, somewhere around about there at the medical school. This shows a picture inside the main brain processing area of the Human Brain Bank, although this only makes up about 1/5 of the space that we have. But really, the Brain Bank is a collection of brains generously donated to advance the treatment of treatment discovery for neurodegenerative diseases. And over the years that I've worked at the Human Brain Bank, I've just been amazed at the generosity of New Zealanders who are willing to offer their brain for future research into brain disorders. So what we have is a prospective donor program. People approach us and sign up for brain donation before they pass away. However, if someone hasn't signed up beforehand, that doesn't preclude the brain donation going ahead necessarily if it's the wish of if it was their wish and the family's wish. We maintain long-term linkages with the donor family or the donor pre-mortem and the family, and then the family post-mortem. And this is a brain bank that's very much research driven. So our processes are changing as it becomes important to change, as new technologies come along and are available. But the key things to point out here is these two hoods here is where we do much of the brain processing. And this is what a -80 freezer looks like, a giant chest freezer. And we have about 10 of those for storing brain tissue. Just to step back a little bit, it's actually in my lifetime that these images were taken using a particular imaging technology that showed that show something that we've known for only just over 100 years, and that is that different regions of the brain do different things. So before that, it was just thought that the brain had a general function and that within the brain, all of our different senses were contained, but not a specific region of the brain that looked after different functions. And so this just shows that when you hear something, there's a couple of regions, two regions that light up in the brain very specifically, other regions don't light up. When you see something, there's two different regions that light up. When you say something, there's 2 regions that light up. And even when you think something, there is a region of the brain that lights up. This makes it very important that if you want to work on hearing, you look at one region of the brain. If you want to work on seeing or what allows us to see what goes wrong when someone loses their sight, you need to look at a different region of the brain. Now, we know this in much more detail than the previous image that I showed you. We know there are some very specific regions of the brain for memory and intelligence and personality, such as the frontal region of the brain here, and for memory and aggression, the temporal region of the brain, and so on. But if you look even at one of these small areas, such as the area that controls our motor movement, different parts of this strip of tissue here will have a different cytoarchitecture. The makeup of the tissue in each of these different regions will differ depending on which part of the body the motor function is being controlled in. So the face region is controlled around here. It will have one makeup of cells, whereas the area that controls the back will have a slightly different makeup of cells. So this means that when we do brain bank a brain, we can't just assume that one piece of brain is the same as the next, quite the opposite. We have to record exactly where each piece of brain tissue gets taken from in order to ensure that when we give researchers a piece of brain tissue, they know what they're working on. And for instance, if it's a neurological disease, that we can give them a piece of brain tissue to study that is affected by that particular disease. So when we receive the donation of a brain, and I'll go over the process of that in a few minutes. When we actually receive a brain, it's important that we dissect the brain in such a way that people can use it, and that the researchers know exactly where the tissue has come from. So when we receive a brain, we first of all cut the brain in half, and the brain gets dealt with, one half being fresh. That is, we don't use any fixatives on the brain, on that half, and the other half we put into a fixative. In fact, we put very small needles into the key arteries, and we infuse the brain with formalin, which cross-links all of the proteins or binds all of the proteins together so that they don't degrade or decay. The fresh side of the brain is useful for doing RNA studies or looking at the native protein state, whereas the fixed side is better for looking at the histology or the cellular makeup of that particular region of the brain. So the first thing we do after cutting the brain in half is to remove the brainstem and cerebellum, this part at the back here, and then that gets divided up into a number of smaller regions that they actually get wrapped in tin foil and labeled with a case number, not the person's name or any identifying details other than a case number, and then also which region of the brain it has come from. And then each of the regions of the brain that contain the motor and sensory part, for instance, get cut down into 4 different regions, and these are labeled 0, 1, 2, and 3. And in that way, we know precisely where that tissue has come from. Then we have the auditory regions, and they get dissected out and preserved very carefully, so we know exactly where that has come from. And each individual gyrus, as we call them, the ridges of the brain, get removed sequentially, and the block of tissue gets recorded in the way that's displayed on this diagram here. So in the end, from every brain that we've received, we are able to locate each specific block and where it's come from. We know exactly where it's come from, so that if you need to compare, for instance, between the superior temporal gyrus block 0 from 5 different cases, then you would be able to do that quite successfully by taking those blocks out of the freezer. This shows the process by which a brain is carefully perfused with formalin so that the proteins get cross-linked and the tissue gets preserved in a way that really preserves it indefinitely, forever. And then from there, each of the small blocks that is cut out of the brain, about typically 1 to 2 centimeters cubed, gets placed onto either tin foil or, in this case, it's a tray. And then this white powder substance, which is actually powdered dry ice, gets shaken onto the tissue, which freezes the tissue very, very precisely and evenly all the way through. Now, once it's frozen, it can be stored indefinitely in the -80 freezer. And much of the vast majority of the tissue that we have gets stored in a -80 freezer, and it really never goes, it's usable forever, indefinitely at that point. And so most of the work that we do requires going into the -80 freezer and taking out the small pieces of brain that we wish to study and working from there. Now, putting the tissue in the -80 freezer is kind of like a fixative in itself. It stops any of the decay or degradation of the tissue. So all of this happens in these in these hoods here, and one of them, this one here, is a chemical hood, so that stops us being exposed to the formalin. And this one here is a biosafety hood, so it keeps all of the airflow in, so that we're not affected by any thing that would be airborne, etc. And then once the processing has happened, then the brains go into the -80 freezer for long-term storage. Now, this means that we have tissue from the original donations that were gifted actually back in the 1980s. This tissue really is something that can contribute to science for a very long period of time. Now, a little bit about how the process actually works. How is it that someone's brain might come to be in the brain bank? Well, first things first, people request a donor pack. And in this donor pack, which we send out either by email or by post, it includes information about what will happen to your brain, what we will do with your brain, or the brain of a loved one, where it will be stored, and all of the details that you need to know about what to do at the time that a loved one dies. And the most important thing about this whole process is actually communicating with your family about what your wishes are. Because when you die, your body actually becomes property of your next of kin or your family. And so ultimately, your wish for brain to donate your own brain comes down to your family wanting to go ahead with that too. So it's important to talk to them about that. And also, if your family don't know that that's your wish, then they're not going to phone us to see that the process can be carried out. So that family communication is really important. Putting it in your will is less critical, and that's because often wills are read after the fact or down the track, and that may be too late at that point for a brain donation to go ahead. We usually have to know that we can receive the brain within 24 hours, otherwise we don't usually proceed. And so that means that gives us one whole day to figure out exactly where the nearest mortuary is, to make sure that all the paperwork's in order, etc., to ensure that the brain can be donated. At the time of a person's death, a family member will call me and or one of our team, Sir Richard Fall, who was the founder of the Brain Bank. They'll either call Sir Richard Fall or myself, and at that point we go through a process of talking to the family member about really just ensuring that this is the wish of all of the family, that nobody's opposed to this idea of brain donation, because at the end of the day, we don't wish to receive a brain from a family where it's going to put them under undue pressure, or where someone feels uncomfortable with the idea of brain donation. It's really important that that there's full agreement to the concept of brain donation. We also need to ascertain whether the person who has died ever raised an overriding objection. This means they knew all of the information about brain donation and they said, no, that's not for me. Because it does occasionally happen that a family want to go ahead no matter what. And it's our job to make sure that we've asked those questions and that it was the wish of the individual and the family. We then organise for the paperwork to be filled out, and the paperwork's actually very simple. Usually people have pre-registered, and so we've got some documentation on file, but this isn't always the case. And so if it's not on file, we would email a copy or fax or get a copy of the paperwork to the next of kin to have a read over, decide whether or not this is for them, and then they sign a form to say that they give an authority for a limited post-mortem examination to be done, which is simply the brain to be removed. We then work with the local mortuary to see that the brain can be removed, and so brain donation can be facilitated from most parts of the country, but outside of the major centres it is more difficult. Nevertheless, we do this on a regular basis to ensure that brain donation is not just for those who live in Auckland, Wellington and Christchurch or Dunedin, for instance. We then work with a funeral director of the family's choosing to make sure that the body can be uplifted and taken to the local mortuary and that all the correct documentation is in place. We also need a cause of death certificate before the brain can be removed, and that's usually organised we can organise that through the local GP. We rely on Air New Zealand to air freight the brain to Auckland if we think we can get it here in a reasonable space of time, and they've always been very, very obliging. It's important to note, though, that we can't always accept, and this is often due to timing, where we just don't think we're going to be able to get the brain to us in a reasonable space of time. Many mortuaries don't operate on the weekend, and so this can mean that the timing can be difficult. We also can't accept if someone had a history of hepatitis, HIV, tuberculosis, or if they've had a very rapid dementia that's not otherwise explained. And this is because it's rare, but prion type diseases can manifest themselves by having a very rapid dementia. After we've received the brain and we've processed it as I've described, we have a neuropathologist who will look through about 17 blocks of the brain that we provide to him, and he makes a formal diagnosis. So this is where, in the months that follow the brain donation, we're able to contact the family again and have a discussion with them about what the results showed. So it's often that somebody may have had an atypical Parkinson's disease, and the pathologist will say, well, this was one of the Parkinson plus disorders, for instance, and we can talk through those results with the family. We also provide written confirmation of the results so that you have something in hand to say what your family member actually had. And one thing just to note here is that we're the custodians of the brain tissue. We're not the owners of the brain tissue. The tissue really, even though it's donated to us, remains property of the family. And so it has on occasion occurred that a family member or family members have down the track thought, actually, this is not for us. We didn't feel comfortable to speak out at the time, but we are now. That really we'd like the brain tissue back, and this can always be facilitated. But we see ourselves as custodians, guardians, if you like, of the brain, not owners of it. And so for those of you who are thinking about brain donation or who are listening because you are a family member of a brain donor, I really want to say thank you, because the amount of research that your donation has facilitated is tremendous. And we couldn't know what we know without people donating their brains to science. So thank you very much. Just to give you an overview of the amount of tissue that we have and the types of tissue that we have, We have approximately 750 total whole brain donations. And this ranges from, age ranges from 1 to 103. Now, the vast majority of the brain tissue that we receive would be from people who are 55 and older, with the average age, of course, the average age gets brought down a lot by having a very small number of very young brain donations, and they are unusual. Unusual in the sense that they're not the most common donation that we get. We have about 37% of our brains are from male donors and 63 from female donors. In terms of the tissue types or the neuropathology types that we have about 227 normal brains. We have 150 Huntington's brains, approximately 73 Parkinson's disease brains or Parkinson's disease with Lewy body dementia brains, about 130 Alzheimer's brains, and so on. So within the other category there are many different brains that are not listed. I'm happy to answer any questions about those if you have them. We also have a process whereby we're able to receive the tissue from people who are undergoing neurosurgical procedures. And so with their consent, we're able to receive tumor tissue or epilepsy tissue from people who are undergoing a resection. And we do actually have about 400 specimens from that particular source. And that's very valuable because we receive the tissue with almost no delay from the neurosurgical operating table through to us receiving the brain tissue. So it's very fresh tissue. So although you might think that seems like a lot of tissue, and so why would you need any more? The reality is that by the time you look at specific age ranges, because you want to match normal with disease tissue for age, for sex, for post-mortem delay, and that sort of thing, it doesn't leave you with large numbers in each of those categories to be able to compare. Besides which, we often use the area of the brain that's worst affected by a particular disease the quickest. And so the brain can become depleted of those areas quickly, and so we're always needing more brain tissue. Now, I'd like to tell you a little bit about what the brain donation can be used for. And I just want to point out what the problem is in a couple of these common neurological diseases. And in the case of Parkinson's disease, we have an accumulation of something called alpha-synuclein. We only recently knew what the chemical entity was that makes up the things that we know as Lewy bodies. And these Lewy bodies accumulate in the nucleus of the cell, of the neurons and other cells, and they also accumulate in the synapse, the connection between two cells, and hence the name syn for synapse and nucleus, or nuclein for nucleus. And what it ultimately causes is a depletion in these large pigmented cells here in the brainstem in the substantia nigra. And in the case of Alzheimer's disease, they have, people with Alzheimer's disease have two different pathologies. One is they have the accumulation of this stuff called beta amyloid or plaques. And this is a sticky substance which fills up the inside of the cell and then ends up spilling over into the outside of the cell. And so this is a cell here, and this is a cell here, and this is a cell here. But all that you see that's brownish, reddish color there is all beta amyloid. So there's a lot of it. Now, this is absolutely characteristic of Alzheimer's disease. However, 40% of people over the age of 40 who don't have Alzheimer's disease will have this in their brain, and 50% of people over 50 and 60% over 60 and so on. So it's quite a common protein. The other protein that you need to get a diagnosis of Alzheimer's disease is these things here called neurofibrillary tangles, or tau. And this tau protein is like the the cytoskeleton, the skeleton inside the cell that keeps the shape of the cell. And what happens is it becomes stiffened so that the cell can't do its normal functions anymore. And with those two things, you end up with Alzheimer's disease. But what's interesting is that even though in Alzheimer's disease, you would normally think that the frontal lobe would be the area that is worst affected because people lose their memory and that's where memory is housed. And the temporal lobe would be affected and people would lose their memory because that's where memory is housed. Sorry, I've just realized that my pointer is not being picked up on the slides. Okay, I'll try and be more specific with what I'm pointing out there. But the frontal lobe there is labeled and the temporal lobe there is labeled. And this is the common places that are affected in Alzheimer's disease. And in the case of Parkinson's disease, the area that is mainly affected is the area under the arrow there, the substantia nigra. But in both of these conditions, 6 to 10 years before the memory loss is evident or the movement disorder is evident, the olfactory bulb, the area that's displayed there with the red arrow, will already have been affected. Now, this olfactory bulb is the area that allows us the sense of smell. And the olfactory bulb is the only area of the brain that is directly exposed to the outside world. So when you breathe in, you breathe odorants in through your nose. They get picked up by the neurons that sit in the roof of your nose, and the scents that are in the air that you breathe in get picked up. Now, they directly influence neurons that are connected to the brain. So this tells us that there might be something about olfaction that, which when it becomes dysfunctional, might indicate that a neurodegenerative disease is on its way. Now, the area just behind those white lines is the olfactory bulb. So you can see the size of the olfactory bulb in the brain on the right is very much larger than the area just behind the white line on the smaller brain, the one in the middle. And the one on the right there is the sheep brain, the one in the middle is a rat brain, and the one on the left there is the human brain. Now, the size of that olfactory bulb in the sheep brain is about, the whole olfactory system is about 20% of the brain weight, and the whole olfactory system in the rodent brain is about 20% of the brain weight. But the brain weight of the olfactory bulb in the humans is about 0.0064% of the brain weight. So it's a very small structure compared to what we have in the sheep and the rodent. Nevertheless, humans do use their sense of smell very exquisitely, but we probably don't rely on it so much as what a sheep does for finding the right type of grass and for its mothering instinct, or what the rodent does for navigating at night and so on. But one of the things that we wanted to do was to understand what was changing in this human olfactory bulb in Parkinson's disease as it became dysfunctional prior to the person becoming Parkinsonian. And so one of the things that we did was to take very thin sections through the olfactory bulb and to stain the key anatomical entities in the olfactory bulb, which are called glomeruli. They're the functional units of olfaction. And each of those small round dots that you can see on this picture is an individual glomerulus. And so we were able to section hundreds of sections through normal and Parkinsonian olfactory bulbs, and then to use some special imaging technology that allowed us just to isolate or to identify the glomeruli, which you can see in the bottom right there. Those red popcorn-looking glomeruli are what we were able to pull out. And then by having a look at, by digitally reconstructing the olfactory bulb, we were able to identify where exactly any deficiencies or deficits there were in these glomeruli or functional units in Parkinson's disease. And so this just gives you an idea of what that looks like. So it's really bringing anatomy alive. It's showing exactly where each of those popcorn, or they look like M&Ms actually, if you'll see in a minute, each of those glomeruli are localized. Now, the textbooks tell us that these glomeruli should be homogeneously distributed across the surface of the olfactory bulb, and they should be perfectly spherical. And yet what you can see here is that they are not spherical. Each of them is different in shape. There are areas of the olfactory bulb that don't have any of them, and areas where there's a high accumulation of them. And from this, we were able to compare normal and olfactory bulbs from people with Parkinson's disease. And we're able to show that in people with Parkinson's disease, they have an accumulation of the red. If you look in B and C, for instance, here, you're able to see some red glomeruli in the middle of the olfactory bulb, which aren't there in the normal olfactory bulb. But also what we found when we looked at this mathematically was that, and if we have a look at the overall global glomerular voxel volume, which is the amount of space that's given over to the glomeruli in normal versus Parkinson's disease, there's about a reduction to about half the normal volume in Parkinson's disease. So there are less functional units in the olfactory system in the human brain in Parkinson's disease compared to normal. Now, the other thing we noted, if we look at the left column there in the graph, is that the distribution of those glomeruli was also substantially changed. So it shows that the undersurface of the olfactory bulb, which is most exposed to the outside world, seems to be worse affected by glomerular loss than the dorsal or the more distant area of the brain in Parkinson's disease. So this made us think maybe there's something in the outside environment, in the air. Maybe it's something to do with the types of colds and flus and things that people get that may predispose them to having this sort of a dysfunction in the olfactory bulb that years later might lead to Parkinson's disease. Now, that's not for certain. We're not saying that that's definitely what's happening. I'm just saying that it's made us think a little bit more about that, and all because of the donation of the human brain and the olfactory bulb, which is part of it, to this type of science. And these are some of the few human studies that have been done on the olfactory bulb. Our ultimate goal here is to be able to identify and treat brain diseases when they are just a smell disorder rather than when they're a memory disorder or when they are a memory or movement disorder. Now, this has also led us to the two, I know my pointer's not working well, but the two kind of raisin-shaped structures in the top of the diagram here are the olfactory bulbs, and the area with the blue blobs underneath it is the mucus-producing cells or glands in the roof of your nose. And all the area around those blue blobs is where the smell is actually first picked up. And so one of the studies that we've been able to work on is to identify exactly what all of the different cell types are or different regions are in that part of the brain, which is probably the area where the brain is most exposed to the outside world. And we can then basically color in the different cell types based on different antibody labels we can use. And then we're able to train the computer to identify all of those regions in all of the sections that that contain exactly that same type of tissue. And so in this way, we don't have to have people beavering away for weeks or months counting different cell types. We can apply computer technology that allows us to pull out far more information than we've ever been able to do before. And hopefully this will also allow us to hone in on what are those initial changes that occur in neurological diseases. Another project that we've been able to work on is to be able to look in the human brain and to see whether or not it is just the neurons in Parkinson's disease, for instance, that are affected by the accumulation of that abnormal protein, alpha-synuclein. So in the brain, we have neurons, which are the main functional unit within the brain. We have astrocytes and pericytes and microglia and a bunch of other cells. And one of the studies that we were able to do was to look at how many different cell types accumulate alpha-synuclein, because we usually think of Parkinson's disease as being a disease of neurons. But maybe it's just that neurons are worse affected by alpha-synuclein than other cell types. And so what we were able to identify was that actually pericytes and astrocytes and microglia actually all contain alpha-synuclein. And by looking at many different Parkinson's disease brains, such as is shown on the right-hand side there, PD52, 53, etc., different case numbers, we're able to see that in all of these brains that we looked at, we could see accumulation of alpha-synuclein, the abnormal protein, in all of these different cell types. But in terms of the large accumulations of them, of the Lewy bodies, the neurons seem to accumulate them a little bit more. But it means that when we look for treatments for these diseases, we shouldn't just be looking at treatments that will affect neurons. We should be looking at treatments that will affect other cell types as well. One of the technologies that we've applied to using human brain tissue is something called tissue microarray. And this, the tray of slides in the foreground there is how we would typically go about doing an experiment. Now, when you add chemicals to each of those slides showing a piece of brain tissue, you start with slide number 1 and you put the solution on. You then go to slide number 2, 3, 4, etc., all the way across the tray, and then you wash them off, going back to slide number 1, and you add the next chemical. And so there's a different amount of time that each of these slides gets incubated in the different chemical solutions that you want to put them into. One of the ways that we've gone about doing things differently is to take very small core biopsies from the tissue that we receive and then arraying them in an array such as that image on the left, lower left, which shows many different cores or different brains arrayed on one tissue microarray. And so this whole tray of slides in the center there can be replaced with just a single slide, which improves the consistency and then also means that we can automate the way that information gets taken from that slide. So some of these ways are how we've gone about making sure that we maximize the usage of the brain tissue that's donated to us, because human brain tissue is a scarce and a rare resource, and we value it very much. One of the things that we've also been able to do, and this is the work of Mike Dragonoff's lab, is to take small amounts of brain tissue and to chop them up into very small pieces, then to use special enzymes to digest those bits of brain tissue so that it's in individual cells rather than in pieces of tissue, and then we can culture those cells to test novel drugs on. So when you die, it's a critical set of cells that have died, but many cells remain alive for actually several days after an individual passes away, and they can be cultured and used for doing experiments on. This just shows some of those cells that have been isolated from a brain and shows them moving around in the dish. This is a time-lapse picture over the course of, I've actually forgotten how long, but maybe a day or so, showing the lives of cells. And then with those, you can see how well the cell can actually exchange material. In this case, it's exchanging some of the cell membrane. The red cell is coming up against the cell that's not red, and exchanging material with it. And these are dynamic processes that you would not be able to watch or observe if it weren't for the donation of human brain tissue. You can see there the cell has ended up with a lot of red particles there, and this is really trying to model the way that one cell can pass on an abnormal protein to the next cell. So I'll leave it there, and just to say a thank you for being interested in brain donation. If you'd like to receive more information, it'd be a pleasure to get you a donor pack or to answer your questions more fully. The best way to get in touch with us is via an email or a phone call, and you can email brainbank at auckland.ac.nz or phone 09923 6072. And it will take us a day or two to get back to you by email, but we'll certainly get back to you. And the most likely person you would hear from is Marika Ashas. But our team consists of Sir Richard Fall, myself, Marika, Remai Parker, Phoebe Anscombe, and our neuropathologist, Dr. Clinton Turner. And really, I'll end before I answer some questions with saying a special thank you to the Neurological Foundation of New Zealand and their supporters for their ongoing support of the Brain Bank. It's very simple. We couldn't do this without you. Thank you very much. I've had a question here around being able to accept people that have had different types of viruses. And I would say the vast majority of viruses that people would have are fine. Most of them resolve, and they're also extremely common. And so we're able to, we do our own serology testing anyway, but if we do know that someone has had hepatitis, HIV, TB, or they've had a very rapid dementia, that we err on the side of caution with those ones. The next question is, does the Brain Bank send material to other labs across the country too? Yes, we do. So we have a process for people applying to use tissue, and that is something that works across the country. So we send tissue to collaborators and people that want to work on human brain tissue at, I think we've sent them to all New Zealand universities over the years, and it just depends on who's doing what research at the time as to who needs that tissue. But it's very much it's the one brain bank, I guess, for the whole country. Do we collaborate with international brain banks as well? And how many brain banks are there in the world? They're two great questions. So each brain bank has a slightly different philosophy and a different way of preserving tissue. So there's probably around about 20 brain banks that would be working on a roughly similar philosophy to the way we run things, where they're research-led and they're constantly trying to push themselves on how much more relevant the tissue can be for different studies that are being undertaken. There are many brain banks that operate as a fixed tissue brain bank, and there the tissue can be made available, but it will only be fixed tissue types.